Good morning, ladies and gentlemen. Myself, Dr. P. Samuel, Assistant Professor of Biotechnology, Ayya, Nadar, Janaki, Ammal College, Sivakasi. Dear students, in this module, we are going to see an important topic from Unit 2 of the elective paper, Biophysics and Instrumentation. The content we are going to discuss is paper chromatography. Right? Let's move on to the topic now. So, in the previous lecture, I have told you that chromatography is a very important technique developed by Michael Sweat in 1900. Right? So, a variant of the chromatography procedure is paper chromatography, which was developed by Singh and Martin in the year 1943. See, while going through important events like year, you should note down it. முக்கியமான 1 mark கொஸ்டின்லாம் பார்த்தீங்க அப்படின்னா இந்த இதில் தான் கேட்பாங்க. ஸோ try to memorize the year 1943. It was developed by Singh and Martin. And as I told you, paper chromatography is an analytical procedure. It is used to analyze complex samples in order to separate the molecule of interest, right? So in general, chromatography is an analytical procedure. Even in many biochemistry laboratories, it is a primary technique to demonstrate the working mechanism of chromatography technique. So wherever you go, wherever you study, right, that doesn't matter. But in a biochemistry laboratory, it is a primary technique used to explain what is chromatography and what are the phases of chromatography, right? So it is an inexpensive procedure. So need of the hour, right? Paper chromatography is an inexpensive procedure. So there are two principles governing the paper chromatography. One is partition chromatography, another one is adsorption chromatography. Right? You should keep it in mind. There are two governing principles. One is partition chromatography, another one is adsorption chromatography. These are the two terms. You should never forget it, right? So what happens in partition chromatography? The sample you have loaded is partitioned between two phases, particularly two liquid phases. I mean, the sample is distributed itself between the water molecules trapped in the pores of the filter paper or mobile phase, which moves over the stationary phase. In sometimes or under some occasions, the chromatography may working on adsorption chromatography principle, which means the sample is adsorbed between solid and liquid phase. I mean the stationary or liquid phase. So you should compare the difference. In partition chromatography, the sample is distributed between two liquid phases. I mean the one called water molecule trapped in the pores, another one called solvent phase. In adsorption chromatography, the sample is adsorbed between solid and liquid phase. So in most of the cases, the principle is partition chromatography, right? In most of the cases, the principle is partition chromatography. So what are all the components? Sometimes the question may be like this: list the components of paper chromatography. So in such case, you have to write there are three basic components of paper chromatography. They are stationary phase, mobile phase, and the sample you are using to separate. Right? These are all the components. Let's see through the diagrams, right? So this is called chromatograph or chromatography jar, right? So in the previous lecture I have given some terms related to chromatography. So take a reference of it or take a note of it, because it will be very useful for you to remember. And the jar is called chromatograph. The jar is called chromatograph because Chromatograph is the apparatus used to perform chromatography, right? So, and this is the one I have mentioned here is called a solvent. So the solvent may be water or any organic solvent, maybe methanol, right? Whatever it is. And I have given a paper strip also, because we are going to use paper, right? The paper is probably made up of cellulose, right? So in paper chromatography, we need to saturate the chromatograph. So what is meant by saturation? இந்த எக்ஸ்பெரிமெண்டல் ப்ரொசீஜர் ஆரம்பிக்கிறதுக்கு முன்னாடி ஒரு 12 ஹவர்ஸ்க்கு முன்னாடியோ இல்லை ஒரு 15 ஹவர்ஸ்க்கு முன்னாடியோ என்ன பண்ணணும் அப்படின்னா, the jar is filled with solvent and it should be closed with a lid. Otherwise solvents are volatile in nature, they may get evaporated. So to prevent it, the mouth of the jar is closed with the lid. So during the 12 hours to 15 hours incubation, the sample, the vapor from the sample covers all over the jar. So this is called saturation. It has to be done in the case of paper chromatography to obtain merit-oriented results, right? See, here I have given two set of diagrams, right? One on the left side and another one on the right side. So left side, it is with the experimental setup, and on the right side you will be having 3 set of samples extracted from 3 different plants, right? All these diagrams are drawn from the tools using in MS PowerPoint, right? So from the samples, it was loaded onto the paper strip, right? See, here I have given 3 different types of sample. Sample 1, sample 2 and sample 3. Right? All the 3 samples are carefully loaded onto the paper strip. Right? So once loaded, what actually happens? The solvent in the chromatographic jar gradually ascends into the paper strip, because there is a force governing the paper chromatography called capillary force. The capillary force allows the solvent to penetrate through the pores in the filter paper. So through the pores, the solvents gradually ascends into the paper strip. While the solvent ascends, the sample also dissolved in the solvent and started to move according to the direction of the solvent, right? Yes, here the sample started to move. Yes, of course, second sample also started to move. Third sample also started to move, and in this way the movement of the sample continues, right? So you can see here that the first migration was initiated in the sample 1, but in the subsequent slide, the migration of the second sample and third sample seems to be much more better when compared to sample 1. And this is because the interaction between the sample and your phases, So as I told you, when the principle is partition chromatography, the sample is distributed between 2 liquid phases, whereas if the principle is adsorption chromatography, the sample gets interacted with solid and liquid phases. Solid phase refers to the immobile phase or stationary phase, and liquid phase refers to the mobile phase. So based upon the interaction or the affinity of the sample with the phases, its migration rate is determined, right? So applications, paper chromatography is especially applied for separating complex mixtures, right? It is used for applying complex mixtures. In biochemistry laboratory, it is used for separating amino acids. Even our own students of ANJAC will have a practical on paper chromatography during their 4th semester, right? They used to separate amino acids by paper chromatography. In diagnostic laboratories, paper chromatography is used to determine organic compounds and biochemicals present in urine, right? In pathologic state, patients are recommended to go to the laboratory for a laboratory test. During that condition also, paper chromatography is playing an important role. Paper chromatography also has a promising role in pharma sector for the analysis of compounds containing hormones and drugs. Even in chemical laboratories, even in basic chemistry laboratories, even in higher secondary schools, we have paper chromatography for evaluating salts and complex salts, right? Let's see the types of chromatography. The first thing is called ascending chromatography. See, the name itself implies that the movement of the molecules are directed towards upwards, and this is because the capillary force leads to the migration of the sample towards the tip of the filter paper, right? Ascending chromatography, the solvent is moving upwards. As a result, the sample loaded in the paper is moving towards the tip of the filter paper. Descending, opposite. Here the solvent reservoir is at the top of the paper strip, right? This is the solvent reservoir. So here you can note down the direction of the arrow mark, right? The movement is downwards. So here the molecules are descending from the top of the filter paper. Ascending, descending. So it is like the solvent moves. The solvent first move upwards and then downwards. Ascending and descending. Combination of the previous one, right? Ascending chromatography and descending chromatography is called ascending descending chromatography, and it will be followed very rare in the laboratory, right? Not as a frequent procedure. Radial chromatography, the one which is given at the center of the paper is called wick, a strip of thread. So this strip is used to conduct the solvent, and the sample is loaded at the center of the paper, and in this assay the sample will be separated in the form of concentric circles, right? Because the solvent moves from the center of the paper towards the periphery. Periphery means the பக்கவாட்டு பகுதிகள்னு சொல்லுவாங்க இல்லையா? ஸோ இதோட மூமெண்ட் பார்த்தீங்கன்னா, சால்வெண்ட்டோட மூமெண்ட் பார்த்தீங்க அப்படின்னா, so from this direction, from the center to the periphery, and your sample is loaded here. ஸோ இந்த ஃபில்டர் பேப்பரில் கேபிலரி ஃபோர்ஸ் மூலியமாக எடுத்துக்கொள்ளப்பட்ட சால்வெண்ட் இந்த சாம்பிளை ரீச் ஆகும்போது the sample dissolves in the solvent and migrates in the periphery direction. And the last one is called two-dimensional chromatography, and this is also an interesting one. And here chromatogram development occurs in two directions at right angles to each other, right? Right. So we'll come to the end of the video. Thank you very much for your patient listening, and this concept is very, very important concept. Paper chromatography is very, very important. Just go through the slides very carefully, and thanks for being with me, and stay tuned with me. I'm sure we will meet in yet another video. Thank you. Bye bye.